I suppose I should say good morning. You've heard a marvelous talk about detailed studies of a disease, atherosclerosis, from Nobuyo. I'm not going to give you a talk about any specific disease, except perhaps the disease of getting old. I'm going to tell you about what has happened in the various stages of my scientific career and what things made me have new ideas. So I should go back quite a long way, as I'm going to be talking about where do ideas come from. So where to start? Well, this is rather a nice little episode that happened to me 2 or 3 years ago. I went back to the school where I was a student from age 5 to 11. I was a student at the school where this plaque is being unveiled. So these are students about the age that I was when I was at that school. And it's rather nice to think about. Let me show you where that school is in relation to where I lived. I lived at that time at this house here, and the school was about 6 kilometers down there. And the school was in a little village called Copley, and the population is quite small, only 1,500 people in the whole population. So how many of you come from a village as small as that? Anybody come from a village of 1,500? Nobody? Oh, well, you all come from a much more densely populated country than the country I came from. But there was a school, and I used to walk from here along the road to the school, but I wanted to see this little river here. There was a rather nice river called the River Calder. When I was there, it was very polluted, unfortunately. But now it's a clean and pretty river again, and you can enjoy it. But there was, at that school, there was one teacher that I remember, and his name was Alan Nethergate. He was a good teacher, and he made us understand a lot. The headmaster, whose name I don't remember, was a terrible teacher. So here I am remembering Alan Nethergate, a good teacher. And then when I was about 11, we moved to the top of the hill about there, just a little bit further than that, to a town now of 100,000. So that's in England, that was quite a big town. And in that town, there was a good high school. In England at that time, some schools were called grammar schools, and they were often related to Queen Elizabeth, but not Queen Elizabeth II, Queen Elizabeth I. And that school was founded in 1585. So it was nearly 500 years old school, and was always there and free. So it was on a street that was called Free School Lane. And there were good teachers there and poor teachers as well. A.D. Phoenix was the chemistry teacher, and he was rather a solid person, and was always worried that we weren't going fast enough. He said, "There's so much to do and so little time to do it in." He was worried, but he was a good teacher. Oddie Brown was a horrible man. Nobody liked him. I didn't like him. And, but he was a marvelous teacher of mathematics. And so he gave me a marvelous feeling for mathematics. I don't think most of the students in the class enjoyed his teaching, but he gave me a feeling for calculus that I still remember with great delight, his teaching of mathematics. In fact, I never had to take another math course, not at the university at all. All the maths that I needed, well, perhaps I needed more, but all that I ever learned was from R.D. Brown. And the Heath Grammar School had a Nobel Prize, me. So they were pleased. Now, I want you to go downstream here on this river. And if we go downstream the River Calder, there's Copley again. Go downstream and you can see the river somewhere here, I guess can just be seen. There is another small town, population 15,000, Elland. And they had a grammar school too, and they had a good teacher, Morris Walsall. And they got a Nobel Prize in chemistry in 1987, John Walker. And he discovered the way that you can generate energy, you can generate ATP from moving hydrogen ions. A marvelous device that he worked out. So go downstream to Elland and you get a Nobel Prize. Now maybe you can go upstream, and this is upstream of the River Calder. There's the river up here, and there's Copley. And you come to another town, not very big town, and it has a grammar school too. And it has a good teacher, Luke Sutherland. And he produced one Nobel laureate in 1951. And another Nobel laureate in 1973. What's the secret? The water? I don't really think so. The teachers, it's clear that it's the teachers that matter, isn't it? So if you become a teacher someplace, you may produce a Nobel Prize, but you'll be important in what you do. So when you're a teacher, you can make a big contribution, or you can learn from a teacher. And these are the teachers I mentioned at these places. And this is my next teacher. This is my tutor, that's my advisor when I was an undergraduate, and also later on the supervisor for my PhD work. And he was in Oxford University at one of the colleges there, and that was an old one. It went back to 1263. So that these are institutions that have been going on for quite a long time, even by Japanese standards. And of course, Japanese culture goes back many centuries. His name was Sandy Oxton. He always liked to be called by his first name, and he was my teacher. And the method of teaching in Oxford at that time, and still is true, is that you're, as I call him, my tutor, the person responsible for your training when you're an undergraduate, would meet with you once a week, and you would read to your tutor an essay on some topic. And for example, you might be told, oh, write me an essay on pain. And that's all you were told. Write me an essay on pain. Well, there's an enormous subject, and you'd have to go away and read a textbook or two, and then reviews, and you were expected to come back with an essay going to the original literature. You couldn't produce a review-type paper. So it was quite a task. And Sandy Oxton had won Nobel laureate in 76, and then me at a later date. But he asked me one time, as my weekly topic, write me an essay, Oliver, on energy metabolism. So that's, again, that's all you're told. And so what to do? Well, obviously you have to go reading things. And this is a book. And you might wonder why I show you a picture of a book. Well, it's a journal, and I remember that journal, exactly what it looked like, exactly what it felt like, what the paper was like, what the typescript was, because in it was a most marvelous article that made sense out of things that seemed to be completely ununderstandable. So you can have, even now it makes my hair stand and then thinking about it, the excitement of reading that particular article in there. I remember where I was sitting, I remember the room, I remember all about it because it was so exciting a thing. So you can be inspired by what you read. And I think when you use your imagination, this must occur to many people in different ways. Perhaps if you're an artist, it's the first time that you see a Picasso original. Or if you're a musician, maybe the first time you hear a Bach motet. Something that goes through you and you never forget. Well, what was in there was an article by Fritz Lippmann, who got the Nobel Prize later on, but not for this particular work. And he invented this symbol. And that symbol means energy-rich phosphate. If you have an organic compound with a phosphate bound in this way, it will have high energy. And the example would be ATP. Now, ATP has 3 phosphates, adenosine, and etc., phosphate, phosphate, phosphate. 2 of those phosphates are energy-rich, and what they have in common is that they are acid anhydrides. Now, that may, some of you have some organic chemistry training, but an acid anhydride is what happens when you take water away from an acid. So if you take acetic acid and remove water molecule, you join 2 acetic acids together and you make an acetic acid anhydride. If you take acetic acid and hydrochloric acid and take away water, you get acetyl chloride. And those are very powerful chemicals that can modify other molecules because they have so much energy in the bond between the 2 acids. And the 2 bonds in ATP, the outer ones, are acid anhydrides, and they are therefore energy-rich phosphate. Now, a phosphate combined with an alcohol is an ester, and an ester is low energy. You don't get a lot of energy from hydrolyzing an ester. And so I came up with this scheme for my weekly essay, and I called it the Smithies cycle. It was before Krebs's cycle, before the tricarboxylic acid cycle had been invented. So I was feeling quite pleased about it, because it would start with inorganic phosphate and make a phosphate ester, and I told you that's low energy, and then by taking away 2 hydrogen, you can convert the low energy phosphate into high energy phosphate bond, because this is now an acid anhydride, and you can generate ATP. And you can go around this cycle, time and time again, converting inorganic phosphate into ATP and get energy for nothing. Well, I wasn't so stupid as to think that it really was true, was correct. I knew it was wrong, but I didn't know why it was wrong. So Sandy and I spent quite a long time working out why you could write this and why it was wrong. And it turned out that the energy that generates ATP depends on the hydrogen, on what's called the chemical protection, chemical potential of the hydrogen. And so we made sense of this, and in fact he wrote an article on this and published it on some thermodynamic and kinetic aspects of metabolic phosphorylation. And he was very kind, he wrote it, but he put my name on it as a scholar of Balliol College. And so that's my first paper, quite a long time ago. But, you know, 1948, about 20 years ago, somebody came up to me and said, "Are you that Smithies who wrote an article in Physiological Reviews?" And I'd never had anybody ask me that. And I said, "Well, yes, sir. As a matter of fact, I am that Smithies." And he said, "Oh, I thought you were dead." So, because it was so long since. But anyway, going on, I did a PhD with Sandy. This is my PhD from my PhD work, and it was, I measured osmotic pressure. It's not important what osmotic pressure measurements are used for, but I was making a machine, an apparatus to measure it, and it was very accurate, because here are my experimental points. And when I published, I had to leave the line, the theoretical line, open in order that my experimental point could show. I was very proud of this. And I published it. But you know, that article has a record. Nobody ever quoted it. Nobody ever referred to it in the literature, except in a list of things people who had done measuring osmotic pressure. And I never used the method again, and nobody else ever used the method again. So you have to say, what was the point of it? Why did you do it? It didn't matter. And the answer is a very important one for students, is that I learned to do good science. It didn't matter what I did in learning to do the good science, but it did matter that I enjoyed it. So I enjoyed doing good science and learned how to do good science. So now if you're doing a PhD, take this comfort. It doesn't matter what you do. It's what you learn how to do good science, not the problem that you solve when you're a PhD, because you don't want to do the same thing that you did for your PhD for the rest of your life. You should do something different. But it is important that you enjoy it. And I want to make you think about that. If you don't enjoy what you're doing in working to a PhD, go to your advisor and say, "I am not enjoying what I am doing. Give me a new problem, please." And if your advisor won't or can't give you a new problem, change your advisor. That sounds like a joke, but it really isn't a joke, because it's so important that you enjoy what you're doing. And if you find after you've got your PhD that you really don't like science, go and become a gardener, or go and play music, or go and write a book, or paint, or do something that you enjoy, because that's what you have to live with the rest of your life, what you want to do. And you can change. Well, this is when I went for my first job. I went to Toronto to look for a job, and I was given a job by Dr. David Scott. And he said, "You can work on anything you like, as long as it has something to do with insulin." And I don't know whether any of you can realize what was the connection between insulin and Toronto, Canada. Does anybody know the connection? None of you know the connection. Well, insulin was discovered in Toronto, and David Scott was one of the first people to work with insulin, and he crystallized it. And his zinc insulin is still used as slow-release insulin to help make it act a longer time. But anyway, he did this. So if you had to do that now, you would probably go to PubMed, and you would look up insulin, and you would very quickly find many things, or you would Google it. Maybe you would Google it and find things. Well, this is Google of that time. These are my planning notes from 1953, and I looked at a thing called the Chemical Abstracts and Quarter Cumulative Medical Insulin listing. And you look for the word insulin, and you got all these articles which would have insulin somewhere in that article. And then you would look at them and find out, well, maybe this might be interesting, or maybe this one might be interesting. And so go on and find your PubMed search. So I did this, and I found that I was suspicious about the method of making insulin, because insulin was made in those days, and still is quite often made in this way when it's extracted from an animal, taking pancreas, beef pancreas or pork pancreas, cow or pig, and extracting it with acid alcohol, with 25% alcohol containing concentrated hydrochloric acid. And I thought, well, no self-respecting protein can stand that sort of condition, so probably there is a precursor. And so I set out to look for a precursor of insulin, thinking that the insulin that was being made was a breakdown product. Well, now there is a precursor to insulin, but I never found it. What I got into was into trouble, because as I started to study insulin, I thought I had better work out a method for looking for a small difference. And I was doing what was called electrophoresis on filter paper at that time. That was you soaked paper, filter paper, in buffer, and put your insulin or whatever you were studying as a place, and then passed an electric current. And this is what happened with insulin. It just unrolled like a carpet. It stuck to the paper. And I was trying all sorts of different things to try to alter this binding, because you can't study something that's just sticking to your electrophoresis material. And then I heard of some work that was going on at the nearby hospital, the Hospital for Sick Children. And they were using a method that had been devised by Kunkel and Slater. And these two scientists made a box and filled it full of starch powder. Now, here in Japan, I probably don't need to talk about starch powder very much, because most of you will have had starch, rice starch. And it's a powder, and you can make fill a box with it and put buffer around the starch powder and pass an electric current, and the proteins will go around the starch grains and migrate. And that's what Kunkel and Slater contributed. Because if they took this is an example, if they took a protein on filter paper, it just unrolled like a carpet, like my insulin. But if they used this starch grains, then their protein migrated as a nice band. But there was a problem with it. And the problem was, in order to find where the protein was, you would have to cut the starch block of grains into 40 slices and do a protein determination on every slice. So imagine one electrophoresis would take you 40 protein determinations. Well, my lab was a very big lab. It consisted of one person, me. No technician, nobody to do dishes. I couldn't do 40 protein determinations to find out where my protein was. But then, where do ideas come from? I remembered helping my mother to do the laundry. What on earth has that got to do? Well, perhaps I should say helping with, in quotation marks. I was there. I'm watching her do the laundry and helping her. And she would take starch powder and boil it and make a slimy mixture, which she would coat on my father's collars and then iron to make a very stiff, nice collar. And when you tied it up at the end of the laundry mixture, the starch is set into a jelly. And I thought, well, I can use that because I can make the starch into a gel. And then I won't have to cut it into many slices. I can just stain it, and I will save 40 protein determinations and be able to go much quicker. And so I went back to the lab, and it was Saturday morning, and I found some starch there, and this is my notebook from that time. And you can see I put starch into this jelly, and there's where it was entered into the starch, and it migrated as a nice clear band. Results very promising. The starch, it didn't bank of the starch, and I could see it by staining. And then, after a little while, just by chance, I tried some serum in the same starch gel. Just as I say, just for a rough test. So I put serum into, oops, that's the wrong one. I put serum into the starch, and this is the pattern I got after a very short run. And I was seeing, it looked like albumin, alpha, I wasn't quite sure what was what, albumin, alpha 1, alpha 2, beta, gamma. In those days, people only thought that there were 5 proteins in plasma: albumin, alpha 1, alpha 2, beta, and gamma globulin. But when I set it up, I must have been working late that night. I set it up at midnight and ran it a longer time, and I got this sort of pattern. Many bands, many more than 5. In fact, I couldn't label them. I didn't know how to label them because it was not clear what they were. But now at least 11 proteins in something where previously there had only been 5. So I knew that I had got onto something interesting, a new method of electrophoresis. And so I went to my boss, David Scott, and I said, can I stop working on insulin and work on plasma? And he was a good scientist. And he said, yes, what you have now found is more interesting than the problem that we were doing together. Start to work on your idea. And my next slide is a sort of summary of what you can hope to happen sometime in your life. It may not happen tomorrow, it may happen 10 years from now, it may never happen. But you hope that if you start with an idea that is your mentor's idea, or your boss's idea, if you like, that sometime while you're in that lab, that you have an idea of your own. And you start to do your idea and slowly stop working on the mentor's idea and you working on your idea now. And you can go out and start a new career with your ideas. But notice one little thing here, that after you left your mentor's lab, you can't stop working entirely. You have to make sure your work is published. And that means when you leave, if you haven't written your paper, you still owe that to your mentor. But if you're fortunate, this will happen to you sometime in your life. And it did happen to me in my first job. You see, not my PhD, not my undergraduate work, not my graduate work, but my PhD work, my first job. And I was getting ready to publish my studies. And here are some samples from two people, George Connell and Gordon Dixon. And you might wonder why I've got these two samples. Well, these guys were my friends, and I got tired of bleeding myself. And so I asked them to give me blood, and this is blood from my friends, Gordon Dixon and George Connell. And you might also wonder why there isn't a photograph. Well, my lab didn't own a camera. That's how primitive it was. Didn't even know, so you had to draw things and make an appointment with a photographer to get a photograph. So I was about ready to have my work published, so I went to, made arrangements to go to have the gel photograph. And just by chance, I ran another sample while I was waiting. I think I'll skip this part. No, maybe I'll backtrack a little bit. But I got out of synchronization here a bit. Let me just backtrack. This then was the Soft Gel Electrophoresis, and I wanted to tell you why it was an interesting system. It was partly accidental. The starch only forms a gel at a high concentration. And when concentrated gels impede the movement of large molecules, so a large molecule can go only slowly through a gel, whereas a little molecule can go quite quickly. So it starts to separate molecules by size. Starch gels therefore separate molecules largely by size. And that's what happens when you use a polyacrylamide. Very quickly after I published this work, people changed from using starch to using polyacrylamide as being more convenient. But it's exactly the same principle. And so it was molecular sieving electrophoresis. And as I told you, it came to me as a result of a childhood memory. And I was given a prize for that, for the discovery and development and application of gel electrophoresis methods that allow the separation and identification of specific proteins. And that was shared with Ed Southern. How many of you have ever done a southern blot? Anybody who's done a southern blot? Yeah, quite a lot of you. Well, this is Ed Southern. And this is all I just said. We both had childhood memories. I told you about mine. The idea came because of helping my mother with the laundry. His idea came from a method that was used when he was a child to make copies of a typewritten document. It was before Xeroxing, and people had used carbon papers. And carbon papers, you typed on the carbon paper, and it would make an imprint onto a paper underneath with the carbon on it. But you could only make 2 or 3 copies, if you were lucky. And a principle was involved mimeographing, where you typed onto a waxed sheet, and it made a hole wherever the typewriter went. So then you put the wax sheet on top of the gel and smeared dye on the top. And the dye would go through the holes, so the dye would now make letters in the gel that are colored. Take off the wax paper and now put a new piece of paper on top and take copies. Southern blots, southern blot, southern blot. So it's exactly the same principle, and again from a childhood memory. So you don't know where ideas are going to come from. I was saying I was ready to publish. And then by chance I ran another sample before finishing. And it was very strange, because there were many extra bands here. Most of it, many extra components. So, BW. What was different about BW, the person whose blood this was? Well, BW stood for Beth Wade. This was a girl. This is the first time that I'd run a sample from a woman. I thought I'd found a new method of telling men from women. So I called this pattern the F pattern, the female pattern, and the other pattern the M pattern for male. And then for every day for about a week, I would do a sample from a man and a sample from a woman, and it always went in the right direction. But then on about the 6th day, they were reversed, and it looked as if the man had changed to a woman. And that's what we asked him, have you changed to a woman? And made all sorts of coarse jokes, which I'm not allowed to tell you about. But you can imagine, come on, let's have a look. But it turned out to have nothing to do with male or femaleness. And here is my data file. But your data file is electronic. But I bet you can't show your data file 60 years later if you have only an electronic copy. You can't even look at a floppy disk anymore. And a CD will become very rapidly unreadable. Make hard copies of your important data. Put the hard copies in a notebook and make sure that you always have something that is physical and can last a long time, that will help you understand when you no longer can read your electronic copy. So anyway, this was showing very quickly that males and females made no difference, and I found a subdivision of the type, and the ordinary blood groups didn't matter, and so it was pretty clear that this was some other sort of thing. And the most likely thing was that it was a genetic difference. And so I went to see this lady here, Norma Ford Walker. It's a long time ago, and she was a white-haired lady, and she was a PhD, as many of you will be, in a hospital. So in the United States, or actually in Canada at that time, or even the whole world, there were very few women who were heads of departments. And to be a head of a department of genetics was even more unusual, because there were almost no departments of genetics anywhere. And she was a PhD, a woman, head of a department in a hospital, a very distinguished lady. And I learned genetics from her. And we've quickly found that the patterns that I had shown you there were due to an inherited difference of a single gene, and the gene was called HB, haptoglobin, because the protein that was differing was a protein that bound hemoglobin, and that has already been discovered and described. So haptoglobin means I bind globin, and this was what that protein was. And then George Connell, whom I showed you a picture of his blood, and Gordon Dixon came back to the University of Toronto as young professors, and we joined together to try to work out what the genetic difference was in the haptoglobin protein. And I won't go through the details except to set the scale, as it were, set the time frame. Nobody could sequence proteins at that time. It was not, nobody had yet invented a method of sequencing proteins. There was a method where you could look at peptides, but you couldn't even sequence the peptides. You could just determine their composition. And George Connell worked out a very good method of purifying the protein, and Gordon Dixon worked out a method of looking at the peptides of the protein, and together we found out that the gene that made the multiple bands it was quite different. It was twice as long. The product, the protein, was nearly twice as long as the product of the gene that made a single band. And we worked out that it was due to what we call non-homologous recombination. Now I'll explain that. Let me represent one copy of the simple gene, A, B, C, D, E, F, G, H, I, and A, B, C, D, E, F, G, H, I, or a slight difference, another copy of the same gene. And what happened was, sometime in evolution of the human species, a crossover occurred to make a molecule which was twice as long. And that was the molecule, that was what the gene was like that made the complicated pattern. And I call it non-homologous recombination because there is nothing in common between GH and BC. That crossover should not occur. DNA in ordinary biology you get crossovers, as Nobuyo explained to you, but they're always accurate. In other words, a crossover will only occur between sequences that are identical. And this was non-homologous, and it turned out to be a completely unpredictable type of crossing over. But once you have a gene that's been duplicated, this gene is here, that's a duplication, CDEF, CDES, CD occurs twice in that gene. Then you can get another sort of crossing over that can occur, and that's where the second CDE lines up with the first CDE of another copy of the gene, and you can make a triple gene. And this is predictable event. It may be not very common, but it's predictable. And we went out and we found some individuals who had the triple-sized gene. So we were able to understand that this type of crossing over is homologous because CDE and CDE are identical, and it's a predictable event. So this work made me understand very well the difference between non-homologous crossing over and homologous crossing over. One is predictable. So as time moved on, we began to be able to work with DNA. And these are the genes controlling hemoglobin. And Jerry Slyton in my lab worked at these, and we isolated these two genes in the hemoglobin series. There are 5 genes that can make beta globin. The adult gene is the beta globin gene, and then there is an epsilon embryonic and 2 fetal genes. And what Jerry found out is that there was evidence of crossing over between these 2 genes. We could see that there were things happening at the DNA level, like the haptoglobin system, that predictably crossing over will occur when 2 sequences are alike. And so that led me to think about correcting the hemoglobin gene, because Linus Pauling and Harvey Itano and Vernon Ingram worked out that the sickle cell gene, the one that causes sickle cell gene anemia, is a change of a single amino acid, a glutamic acid to a valine. So that this is the globin gene of sickle cell, and this is the globin gene normally. And I thought that, well, maybe we could use then a homologous recombination to correct that gene. And so this is a globin gene, the wrong one, and here is a piece of DNA that is a normal sequence, and I can get homologous recombination, SA crossing over and GEN crossing over, and I can repair the incorrect gene by homologous crossing over. And that began to go through my head as being something that it would be nice to be able to do. But I didn't know how to do it. But I was teaching. And I read this article. It came out in April 1st of 1982. And I had to teach this in my course. My course was always up to date, and I read about this paper, and it was a paper in which, without going into a lot of detail, in which the authors used a method of looking for two things coming together in order to isolate a transforming gene. That's a gene that will make a cell change from normal growth into abnormal growth, like a tumor-forming gene, if you like. And they used a method where they had a piece of DNA that they could recognize, a blue piece of DNA, and they were going to look for the red piece of DNA as their transforming gene. Without going into detail, it was blue coming next to red, and they had a method of doing that. And I thought, I can use this method to test whether gene targeting is possible. So this is from my notebook, only 3 weeks after that paper came out, Thursday, April 22nd, and in which, of all of my notebook pages, this one is the one that I'm most proud of, because in it is how to do gene targeting and how to get a Nobel Prize, if you like. Because it says the aim is to place the correcting DNA in the right place. In other words, gene targeting. I didn't call it that. The need is an assay. And here is the possible assay. And the assay consisted of having a blue piece of DNA, the same one that I learned from the teaching. Take the blue piece of DNA on the incoming DNA and look for it coming next to the target gene, the red piece of DNA, which is the beta-globin gene. So if I do an experiment and I can find a piece of DNA where blue is next to red, I will have proven that I have hit the gene. Because that blue and this piece of DNA can only be produced by this construct targeting correctly. It was quite a difficult experiment, and you had to make a targeting construct. And even that was difficult. Here, 2 pages from my notebook. I really can't tell you what they are anymore, but it's quite pretty. But anyway, eventually we got to the stage of having now a piece of DNA to use in this experiment. And I was collaborating with Raju Kuchulapati, who was in Chicago. So I would send him the DNA that was to be used in the experiment. He would do the experiment. He would isolate cells in which the gene had been introduced, and then make DNA from them and send the DNA back to me to try to see if I could find blue next to red. And it was a complicated and very cumbersome assay. But eventually we got to the stage of testing it, and here was from Raju, and here was the experiment. And I happened to notice the date of the day. It was June 23rd. Well, why is that special? Well, it's my birthday. So that was the first time we did a test. It was on my 58th birthday. It didn't work. You don't get special results on your birthday, but you can enjoy doing it on your birthday. Anyway, eventually we simplified the method, and this was the way we would do it in the simpler version. So here is the beta-globin gene, and here is the targeting construct, slightly different from the old one. But if we did a southern blot, we would find, if the gene had not been altered, an 11 kilobase fragment. If the gene had been correctly altered, we would find an 8 kilobase fragment. And we went through the experiments and got to the stage of testing it and looking for the recombinant fragment. And here is the result, the first time that anybody has seen gene targeting, because this is the first time when the band has changed. So the southern block was from different colonies, and here are 10 or so colonies which have not been targeted, and this one has changed to be the right size, the 8 kilobase. So this is the first gene targeting experiment to work. It took 3 years from the beginning. I now think that that was quite quick, really. It may seem a long time, but 3 years isn't so bad to get from trying to do gene targeting to having proof that it worked. And we published together. And I only want to read one little statement in here, that although the frequency of success, it was really about 1 in a million of the cells treated, is at present modest, the data show unequivocally that the planned modification of a specific human gene is possible. So this showed that gene targeting was possible. And once you know something is possible, many people will then try, because they know it's possible. And that they don't have to say, well, maybe it won't ever happen. And so, but it was clearly no use for treating patients. 1 in a million successes, that was no use. So what to use it for? And then I heard about the embryonic stem cells, and you all know what embryonic stem cells are, but here's an example of colonies of embryonic stem cells from a mouse, 129 mouse, with yellow fur, and these are colonies of embryonic stem cells. And they were put into an egg from a black 6, a black furred animal, and they join and make a hybrid animal, or really not a hybrid, a chimeric animal. And this is the chimeras, which are part of the constitution, comes from the embryonic stem cells, and part come from the the embryo into which the cells are introduced, and you get the gene targeting to work. So I needed embryonic stem cells. What to do? Well, go and ask Martin Evans for them. Martin Evans had these cells, and he came to, he was visiting the United States, and he came into the lab one day, said, "Hi, Oliver," and reached in his pocket and said, "Here they are." He brought the cell in his shirt pocket. And this is an example of what science should be like. He didn't ask for any conditions, he just gave them. So that's what science is, sharing. And if you learn to share, you will enjoy the other side. You will enjoy the happiness of sharing. And I say that making your science available to other people will give you 9 times out of 10 you will be happy. 1 time out of 10 the person who gets them may cheat on you or not acknowledge you. But 9 successes for 1 failure is a pretty good deal. So help people and you will be helped in science. Oh well, what was this about? Well, this was saying that Mario Capecchi also went to see Martin Evans. And we both went, quite surprisingly, at the same time. Within 3 weeks, by chance, Mario Capecchi was also working on gene targeting, and we both went and got help from Martin Evans. But remember how laborious I said my assay was. And then I heard Kerry Mullis talk. And how many of you know who Kerry Mullis is? How many? How many of you know what PCR is? Polymerase chain reaction. Well, you ought to know Kerry Mullis, because Kerry Mullis is the person who invented PCR. And I heard him talk, and I thought that I can use his method to find out whether gene targeting is working, instead of the laborious method that I had devised. And so I began to use PCR. But it was before you could buy a PCR machine. You couldn't buy one, nobody had yet begun to manufacture it. So here is my PCR machine. 3 old water baths, and here it was all sorts of pipes. In fact, we called it a hexapus. You know what an octopus is. It has 8 tentacles. This is a hexapus. It has 6 tentacles, each 2 going into one water bath. And I think it ought to have this label on it. That was a label that used to occur in the lab where I was doing my PhD in Sandy Oxton's lab. You would, every now and then, you would find some piece of equipment with this label on it. I'm going to translate it. It says, "No bloody good, but okay for Oliver." But my friends would know that I would use junk, you see. So this apparatus was made out of junk. These were old water baths that I found in various places, and so they are NVG boxful. Well, you've heard how marvelous the results can be got with gene targeting from Nobuyo. Here is a lovely animal with the atherosclerosis. I'm proud not to be an author on this paper. You see, I'm not an author. I didn't think about it. I didn't do it. Nobuyo did it. And you can be proud of your wife, I hope, or your husband. Well, what's he doing? Well, I'll quickly tell you what he is doing, because notice now I put his date of birth and date of dying. He asked me when he was about 75, he said, "Oliver, when I die, I would like you to write my memoirs, my scientific memoirs." Scientific memoirs were needed in a journal. And I said, "Well, I don't know whether I'll die before you or you will die before me. But if you die first, I will write your memoirs. And if you're going to write the memoirs of somebody, you have to read their papers." And he had a lot of papers. And I read all his papers. And one of them was a very pretty paper with this formula in it. I'm not going to tell you much about it, except to say that it has something to do with the space in gels, and molecules of radius big R and molecules of little r. And I liked the formula. And then, so it was in my head, this formula. And then that's what it means, that if you have a gel, and this is a gel fiber, and little molecules can find lots of space in the gel, big molecules can only find a little space. So big molecules are less common in a gel than little molecules. And that was his principle. And then I went to a meeting in Stockholm, this is before the Nobel Prize, and I gave my talk. And then I heard this guy talk about the kidney. And he described the kidney and how it might work. And I realized that maybe the kidney worked by what I might call the Oxton Principle. And that this is the glomerular basement membrane, and that small molecules would come out of the basement membrane at the same concentration as in plasma, that big molecules would come out at the concentration much reduced by that gel effect. And I published a paper on it, and I'm not going to spend much time on it, except to say that in order to test it, I thought I would use gold nanoparticles, because I could make gold nanoparticles of different sizes. And where does your inspiration come from? Where do the ideas come from? Well, it turned out that Michael Faraday, back in 1857, had been making gold nanoparticles. He didn't know they were gold nanoparticles, but he did know they were beautiful. And he said here that agents competent to reduce gold from its solution are very numerous. And he used phosphorus. If the solution be weak and the phosphorus clean, part of the gold is reduced into exceedingly fine particles, which becoming diffuse produce a beautiful ruby fluid. So I tried his method, or a variant of it, and there you see the colour of these particles. A beautiful red. They are very stable. They're so stable you can make glass with them. And if you go to Europe and you look at some of the stained glass in some of the churches there, and it's gold particles, gold nanoparticles that are in the glass. Anyway, I learned to make them and found methods of making them different sizes. To give you an idea of the scale, these are some particles that are about the size of an immunoglobulin molecule here. That's an immunoglobulin molecule. And here's a very small crystal of them. These are individual gold atoms that you can see here, and there are about 1,000 gold atoms in that little crystal. And I've used it to study the kidney. And here are some big particles in plasma. Here is the basement membrane, and you can see that there are fewer particles in the basement membrane than there are in the plasma. So there is some Oxton effect here. Oh, what's this one? Oh, well, that's 2014. And it's Saturday. I still work weekend. And I got a paper out of it. I'm very proud of this because this is the first time I published a paper in a chemical journal. And to have a first author paper, I'm the first author on this paper, and quite properly I did most of the work. And I wrote it. To have a paper when you're 89, you can feel pretty good. One last thing, and then I'm going to stop. And that's this. That's a person who influenced me a great deal. His name is Phil Morrow. He taught me to fly. And here I am with him in 1980. And we were about to set off on a trip across the Atlantic, and we flew across the Atlantic in this airplane, and we got a world record for that size airplane flying across the Atlantic. But like nearly all people who learn to fly when they start quite old, I was 50 when I started to learn to fly, and you're nervous and sweat. I would sweat so much that it would drip from me. And I remember turning to him at one stage and saying, "That was a good day. Only one drop dripped." In other words, normally many drops drip, but one day only one drop dripped. And then I realized what it is. That by the time that you're nervous and frightened, and you overcome fear with knowledge. And so Phil Morrow taught me to overcome a fear of flying. And that when I became an instructor, I found exactly the same thing. I had this student, Jeff Block, and I was teaching him to fly a glider. And the student is always in front of you, and you're behind in this situation. And he would come back from the lesson absolutely, his shirt would be completely wet, absolutely sopping wet. When he went by himself, on the first time when a pilot goes by him or herself is the solo flight. When he went back and did his first solo flight, he came to me and he came up and he said, "Look, Oliver, dry." Think what that means. He'd learned enough now that he could do something that he used to be frightened of, and now do it and not sweat because he's no longer frightened. The same thing is true in science. You think this person over here is doing some work that I would like to do, but I'm not as smart as she is, and I couldn't do it. or it's too difficult. That's fear of doing something new. Go and learn. Go to the person and say, teach me. Teach me how to do it. And overcome your fear of something new with knowledge. And that will be one of the most important things that you can learn. If you learn that there's nothing that you can't do if you learn about it. Go and be taught. It's a good thing to have a hobby. That's my airplane, and I still fly it. And marvellous to have a companion. And there's my companion over there. And that's where I'll stop.